Hello, students. Hope that you're good. Today, I'm going to tell you about the adaptive immune response. Uh, as compared to the innate immune response, we know more about the adaptive immune response. And you know that the major cells that are playing their roles in adaptive immune response are the T cells and B cells. So, in this presentation, I would talk majorly about the B cells, and then later on, I would be talking about the T cells. Um, the B cells are important because these cells are making the weaponry to encounter the pathogens, and that weaponry actually is the antibody that the B cell would produce. And the T cell is performing different functions. There are different effector types of the T cells, like there are memory T cells, there are cytotoxic T cells, there are helper T cells. So they are doing different jobs. And one of the T cell uh, personality, which is actually the helper T cells or CD4 positive T cell, is helping the B cells with this process of sensitization and antibodies production by the B cells to turn into a plasma B cells and then produce antibodies. And the antibodies then can be used in different ways to neutralize or clear off the pathogens from your body. For instance, the antibodies can be used for opsonization, complements activations. They can be used for ADCC or antibody-dependent cellular cytotoxicity. So there are different ways that this antibody can be used to get rid of the pathogens from inside the body. And that happens when the innate immune response has already failed, and now it is the turn of the adaptive immune response to take care of this resistant pathogen that has just crossed the barrier of the innate immune responses, and now it is here. So in the picture, you see this is a resting B cell that is shown here. And there are some spikes on the surface of B cells that you can see. These are the B cell receptor. So on the surface of a resting B cell, these B cell receptors, one B cell can only have one type of B cell receptors. A B cell cannot have many different types of receptors on its surface, uh, like many different types of B cell receptors cannot be there because one B cell or one T cell, they are specific for one type of pathogens. And how many different types of pathogens can be recognized with the help of these B cells? So, you can just look at this antibody repertoire that 10 raised to the power 9, it's a huge figure. The total number of different specific antibodies that can be made by an individual, that number is about 10 raised to power 9, which is a huge number. We don't know about so many different types of pathogens, but we know that there is a capacity in the B cell to produce so many different types of antibodies or to do so many different types of rearrangements of their genes to produce these different molecules or the different peptides expressed on their surfaces. So in the first frame, you see this B cell with these B cell receptors on top, and all of them are of similar types. You can see that all of them are yellow receptor. And this resting B cell, the surface receptor of the resting B cell, which is the B cell receptor, it's also called immunoglobulin. These are globular proteins, and these are also glycosylated protein. It means glucose moieties are attached to these proteins as well. We call them glycosylated proteins. So these B cell receptors can on the surface of a resting B cell can either be IgM or these can be IgD as well. And class switching of antibodies, there are different classes, you know about the 5 different classes of antibodies. But if you take the example of a resting B cell, so the antibody which is expressed on the surface of a resting B cell can either be IgM or IgD. And later on, the class switching takes place, and that is for a purpose that I would tell you later. In the second frame or in the middle, you can see that this B cell has found its perfect partner. This is a bacterium. In this bacterium, the epitopes or the antigens or PAMs expressed on the surface of these antigens expressed on the surface of this particular bacterium, uh, you know, have been recognized by the B cell here. So this B cell, it means, you know, is now bound to its specific antigen, and this B cell is required in greater number inside the body. So first, where does this encounter take place? The site of the encounter is also very important. uh, this encounter is not taking place everywhere in the body, although you see that antibodies are everywhere in the body, but the B cells are not, all around in different tissues of your body. This is a specific site for this kind of interaction between an antigen and antibody. And that happens in the lymph nodes. These B cells and T cells are majorly found inside the lymphatic system, inside the lymph node. And this interaction of the antigen with the B cell takes place inside the lymph nodes. So if you want to, find out an antigen, a bacterium or a virus has entered the body, and now this bacterium and the virus has to uh, you know, has crossed the innate immune response, and now the dendritic cell has taken these antigens inside the lymphatics, and in the lymphatics, these antigens would now be introduced to the B cells and T cells, which are residing there. That is the home of the B cells and T cells. That is the place where this interaction would take place. And the antigen because there are many different types of B cells there in the lymph nodes. There are many different types of T cells in the lymph node. So the antigens which have been brought to the lymphatic nodes by the dendritic cells would now uh, be matched with different B cell receptors and different T cell receptors. And out of that, large population of B cells and T cells with different B cells and T cell receptors, the antigen would find out its perfect partner and would bind to that particular B cell. It means that particular B cell is required, and now this B cell has to be expanded in number because other B cells are not required at the moment. They do not fit with the antigen, so the body's mechanisms are that the B cell has to be expanded. So clonal expansion of this particular, B cell is required so that the number of these B cells would increase, and these cells would encounter because these are the specific weapons against that particular antigen or that particular bacterium which has entered in your body and which has crossed the innate immune response and now adaptive immune response is active against it. So that's why this B cell is required and this needs expansion so that this weapon should be, produced at a greater, or you can say, a massive level. So in the next frame, you see that stimulated B cell gave rise to antibody secreting plasma cells. These plasma cells are also B cells, and they are secreting the same type of antibody that you see here in the middle frame and in the first frame, which was expressed on the surface of the B cell and which encountered with the antigen. So this B cell, uh, the plasma B cell, after this interaction, certain genetic changes would take place inside the B cells that we would discuss later. That what are those changes? And these B cells would be turned into an effector B cell, which produces these antibodies. These immunoglobulins expressed on the surface are the B cell receptor, which was expressed on the surface of the B cells, would now be produced by these plasma cells, and they would be secreted out. And once these antibodies are secreted out, you can find these antibodies in different tissues of your body. They are in the lymph, they are in mucosal surfaces. These antibodies are in the blood, and wherever they would find these antigens, they would just go there and bind to them. So they can do labeling of the antigens for different purposes, like for activation of the complements, for antibody-dependent cellular cytotoxicity. They can enhance opsonization or phagocytosis of the antigens when they label it. Then it becomes easier for the cells of the innate immune system to identify the pathogens where the antibodies are attached and to just engulf them. So what actually is an what is the structure of an antibody? That is also very important. In the next slide, I would just describe about the structure and what are the important domains in the antibodies and what is class switching. So in the next lecture, we would discuss that.